Hello, everyone, and welcome to today's webinar titled Panel Presentation, Overcoming Challenges of Copy Number Variant Interpretations with QCI Interpret and the ACMG Guidelines for CNVs. This webinar is part of the Clinical Diagnostics and Research event, and I'm Susie Valdez of LabRoots, and I will be your moderator for today's event. Today's educational web seminar is presented by LabRoots and sponsored by Kaijan. For more information about our sponsor, please visit them at QIAGEN.com. So let's get started. Before we begin, I want to remind everyone that this event is interactive, and we encourage you to first participate by communicating with other attendees using our new live chat feature used during the presentation. You can find that live chat feature located at the right of your screen. You can also participate by submitting as many questions as you want during this presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. If you have any trouble seeing or hearing this presentation, just click on the Help Desk button located at the bottom of your screen where the navigation bar is and from the lobby. And finally, as a reminder, the presentation is educational and offers free continuing education credits. Click on the Continuing Education Credits tab located at the abstract window below the presentation window and follow the process to obtain those credits. I now present today's speakers, Dr. Dan Richards, the Vice President of Global Clinical Product Management at QIAGEN Digital Insights, and Dr. Martin Jones, the Scientific Product Owner at QIAGEN Digital Insights. For a complete biography of our presenters, please visit the Biography tab at the left of your screen. Dr. Richards, Dr. Jones, welcome. You may now begin your presentation.
Hello, everyone. I would like to welcome you to the QIAGEN Digital Insights webinar on Overcoming Challenges of Copy Number Variation Interpretation with QCI Interpret. My name is Marianne, and I will be the host and moderator for today's webinar. Now, I would like to introduce our speakers for today, Dr. Martin Jones and Dr. Dan Richards. Dr. Martin Jones is a global product owner for clinical decision support software and interpretation services at QIAGEN Digital Insights. Dr. Dan Richards is the Vice President of Global Clinical Product Management for QIAGEN Digital Insights, where he focuses on the automated interpretation of molecular variation in the context of current clinical evidence. Thank you for joining us today, Martin and Dan. I'll now hand the presentation over to you.
Thanks. So I'm Dan Richards. I head up clinical product management and touch on some of the capabilities our QIAGEN Clinical Insight Interpret product can provide to help overcome the challenges of copy number variation interpretation. I'd like to start with just a brief introduction to QIAGEN Digital Insights. That's a group of several hundred scientists, as well as hundreds of engineers and other specialists that work daily to keep on top of the new clinical literature and provide products that help translate genomic data into clinical insights or actionable insights. We have a variety of products in different areas, from academic research on human samples all the way to clinical laboratory testing. And one of the core things we do is curate this evidence from the scientific literature, keep it up to date for you so you can get a constant current view of what's known to be clinically relevant and gain insights into your data. We have over 90,000 customers worldwide. Our product's been cited over 35,000 publications, and the particular product I'll talk about today, QIAGEN Clinical Insight Interpret, has been used to interpret over 2 million patient samples to date. Since this knowledge base that we create is so core to our products and what I'll talk about today, I just want to touch a little bit on our process. So our curation is sort of like a factory-like process. It's high scale. We have hundreds of scientists working on the curation. It's systematic. It goes through curation protocols and processes that are rigorously tested and defined. And that provides very high quality computable evidence that then can be used to put the data that is generated from next generation sequencing, for example, into context of what's currently known about the variants or alterations detected, for example. We curate a variety of different sources, of course, scientific literature, all the major journals, but we also curate medical guidelines like practice guidelines, drug labels, integrate various other databases that are commonly used, as well as molecular data sets in clinical trials as well. So a wide variety of different content sources that are clinically relevant that we curate on a daily basis. And that is done through a distributed workforce of scientists that are trained on the various protocols we use and read the literature or review the other sources and integrate it into our what we call the Kaijan knowledge base, which then enables our various applications to compute over it and take data that you may generate and put it into biological context for you. And so over the past 20 years, we've curated over 20 million findings, as we call them. I'll just show an example of what that is. It's sort of everything we curate is fully transparently tied back to the source that we curated it from. So in this case, this is some example, what we call variant findings from this particular article that show various details in terms of whether mutation is germline, what the translation is, the disease association, the population observed with the mutation, the variation, whether it's childhood, adult, a wide variety of details that when they're present are captured in our curation process. And this evidence enables us to map, for example, this finding if it appears in a sample you run through Kaijan Clinical Insight, for instance, to be matched to it and provide insight into case reports, for example. So Kaijan Clinical Insight Interpret, if you're first time hearing about it, I just want to give a brief introduction to it as well. It's our premier clinical interpretation and reporting tool. It's an application that's web-based, so you sort of upload your data through APIs or user interface into QCI. And then you analyze it if you're doing translational research, or if you're a clinical lab doing a laboratory developed test, you can use it for clinical decision support and sort of review the variants in a test in the context of QCI. What's core to it is it's evidence-based software. It takes those variant findings, that evidence we curate, I mentioned, into account computationally, and then assists in your review of the alterations or variants you detect in any particular sample to enable you to assess which ones are clinically significant. And if you're using a clinical decision support version of QCI Interpret, report out, create final reports as needed. So core to what we provide is this comprehensive annotation and literature resource. We have all kinds of information about case reports and functional studies, frequency in the general population, and in a somatic cancer context, this is also applicable. It helps identify what treatments may be available, clinical trials are currently available in a particular location, prognosis, and a variety of other information as well. So we really try to provide a comprehensive view for human samples, which variants are clinically actionable and in what ways too. And this is done in a very scalable, automated way. So we provide out-of-the-box alignment of our content with professional guidelines. I'll touch on that a little bit more here, but it basically enables all the variants you detect to immediately get dynamically classified based on the ACMG guidelines for germline variants or the AMP guidelines for cancer or somatic variants, so that you can immediately identify which ones are perhaps most clinically relevant to review and potentially include on a report. And that it can be done at scale, since we cover all the commonly clinically tested genes for hereditary disease as well as cancer, as well as have full HGMD integration within QCI Interpret, sort of cover sort of the whole exome in our ability to put variants into biological context and provide insight into which are potentially disease-causing or disease-promoting. For cancer workflows, we also provide actionability in terms of defining which treatments and clinical trials given the patient's gender and age and other considerations, diagnosis of tumor are relevant for that particular case right out front automatically. And we provide customized reporting too. So some of our customers, they have their own, they use QCI fully for producing final signed reports. There's a whole workflow support integration as well in terms of multiple levels of review if desired, and lab director sign out with digital signature, support full LIMS integration as well through APIs and too. So the whole product over here, just to put it in the context, is really focused on not secondary analysis, but for the interpretation reporting part of the overall workflow for genomic data interpretation, whether it be germline or cancer somatic samples that you're reviewing. So briefly, again, what we provide with our curated evidence is the ingredients, so to speak, for assessing the clinical significance of variants. And we assess the clinical significance of variants in the germline context using the AMP guidelines, which puts every variant in a category from benign to pathogenic. For somatic cancer variants, we also have the actionability aspect, which provides a tiering in terms of, for that particular mutation, that particular diagnosis, tumor type, you know, how actionable is it for a patient in terms of treatment trials or prognostic significance, for instance. So again, all this information we constantly curate on a daily basis, and the knowledge base powers the ability for us to apply algorithms that use these guidelines as frameworks to automatically provide an up-to-date computed classification in both these senses for your variants that you detect and having your samples that you upload into the system, for example, in variant call file format, and lets you quickly triage them and identify which may be relevant for your review or reporting purposes. So I want to touch now on copy number variation overall, and sort of dig deeper into what QCI does, particularly for copy number variation. There's quite a variety of things. That gave you a quick flavor of some of the things it does, but today's focus really is on what QCI provides to assist in assessing copy number variation. So just putting things in perspective, the whole field of cytogenetics has evolved quite a bit over the last 20 years, from FISH to chromosomal microarray or array comparative genomic hybridization approaches, and now a lot of folks are using NGS for detecting copy number aberrations. The reason NGS is particularly useful is it can detect things that are small copy number variations. So by copy number variation, I mean both deletions of part of a chromosomal region or of a chromosomal region, like either exon level deletion or a whole gene deletion, or it could be a large chunk of a chromosome arm, a cytogenic deletion as well. NGS can help detect all of those types of chromosomal aberrations. It has some advantages over MLPA, sort of PCR-based, for example, methods, since it can be applied to many genes in a panel or all the exons of many genes in a panel at once, whereas MLPA is more tailored to particular exons. It's not that scalable, so many labs use NGS, and if there's a copy number detection in NGS, can always confirm with MLPA, for example. And likewise, relative to the older chromosomal microarray, NGS panels sort of provide a deeper level of resolution than chromosomal microarray can, unless you have a very customized design. So NGS avoids the need to create a lot of customized probes, and there's a limitation of probe placement for microarrays, of course, based on which variants are polymorphic in the general population. So NGS provides much more data. It can provide very precise endpoints of deletions or duplications of genes. And that's valuable because it increases the diagnostic yield that the assay can provide. So not only can it provide a simpler workflow than using MLPA, for example, on a routine basis, it also can provide a more simpler and cost-effective workflow with a higher diagnostic yield, which is what we all want for our patients' benefit. So the American College of Medical Genetics recently provided new guidelines to interpret copy number variation. So, both for cytogenetic laboratories that work sort of with chromosomal microarray and RNA-CGH, among other methods, on a routine basis. Many laboratories have also adopted next-generation sequencing, and both of them, since they both can detect copy number changes, want to make sure there's consistency in terms of how those copy number changes are clinically assessed. And so through a wide variety of participants, the American College of Medical Genetics and Genomics, and also with ClinGen funded research, sort of got folks together to identify how to go about, in a consistent way, assessing the clinical significance of copy number variants. And they came up with these guidelines, which are intended to be used both for cytogenetic or array-CGH chromosome microarray detected CNVs, as well as for NGS CNVs, to try to sort of standardize interpretation in the field. Along with that publication, they provide this, as you see on the right here, what's called the ClinGen Cmv Pathogenicity Calculator, which, if you're not familiar with these guidelines, can give you a sense of, there's about 80 or so different considerations or questions that it goes through to help determine a final assessment. The overall assessment ends up in the same ACMG framework from benign to pathogenic. So you can get pathogenic, likely pathogenic, plus, or likely benign or benign, of course. And these guidelines are for hereditary or germline CNV interpretation only, just to be clear. So what does QCI Interpret do for copy number variant interpretation? There's many things, but there's sort of three things I want to highlight today. One is to provide supporting literature content. We have a bibliography feature in this application, and that supporting literature content addresses the need of determining whether a similar CNV has been reported in another patient in the literature. So the need here is, really understanding whenever you get one of these copy number variants, without matching them to some other evidence that they've been seen in another patient, or having some really strong first principles reason, such as a full gene deletion, that the copy number variant is going to affect function. It's good to have the supporting literature to understand, is there a case in the literature which has an overlapping very similar CNV? Typically, they're not going to be identical. And if so, does that patient or those patients reported in the literature, do they have similar symptoms with the patient you may be assessing the sample of at hand? The second area is this new ACMG CNV guidelines, sort of automatically computing pathogenicity based on them. And this is more of a triage need. If you're doing exome or whole genome, which are a translational version of QCI Interpret, many of the customers use sort of exomes and genomes, you have this need where you have your pipeline may detect many copy number variations at the smaller exon level scale, and whether they're exon deletions or partial gene deletions or amplifications, duplications of gene regions, it can be challenging to figure out which of those are most likely pathogenic. And so the guidelines give a principled way to automatically classify all those based on the current evidence and knowledge we have about those genes, so that when you look at your sample with these many CMV variants called, you immediately can see the ones that appear to be likely pathogenic or pathogenic at the top of the list of variants, for example, when you look at a sample or a case in QCI Interpret. And the third area is, aside from the published literature, which we curate, the ACMG guidelines, which we dynamically compute and provide to triage your variants, is the ability to link to unpublished data. So there's a variety of excellent databases out there that have gathered copy number variant reports, some in the literature, but often many not, such as Decipher, that you want to that we provide the ability to link out to review the evidence, to look at the cases in those external databases, to understand likewise, is a similar CNV in a patient, does it exist, and what's known about that patient, even if they're not in the published literature we curate. So I'll go through each of these in a little more detail, but sort of wanted to start with the highlights. And also, before getting into more detail there, just touch a little bit on the workflow so we're all oriented. In particular, what we provide with QCI Interpret, and the translational is the academic research edition. The other editions are for clinical decision support and laboratory developed test purposes, is a flexible and scalable evidence-based variant interpretation platform. So it is focused primarily on variants from next-generation sequencing, although it can be adapted to microarray CMVs as well. But whether you use a Kaijan QIAGEN panel or any vendor's assay as chemistry, it sort of supports them all. Kaijan Digital Insights offers a product called CLC Genomics Workbench, which has nice integration with QCI Interpret, does the secondary analysis of any panel, so to speak, or genome or exome assay, and translates it into the variants and copy number alterations as well that are detected in the sample. But you can use any secondary analysis with QCI Interpret, so sort of support all vendors and public pipelines through the standard of the variant call file format, which is the input into QCI Interpret, along with some metadata about the sample, either entered through the UI or sent along with the VCF through the APIs. So then QCI Interpret does this filtering, it does the variant classification, helps put things in the biological context, and can get you to that final report or export from an API to integrate to your LIMS to produce your own report. So along this workflow, I just give an example here for everybody, your edification, is that there's the CLC Genomics Workbench has this tool that detects copy number variation. One way to think about it is generally the way informatically these copy number variants are detected is you have a set of baseline samples which you believe are normal, are diploid 2 chromosome copies for each sample, sequence a bunch of those in your environment, and then you use that as a baseline to then train the software here, in this case Genomics Workbench, to identify what is a statistically significant decrease, which would be a copy number loss or a region of the genome that's deleted, or statistically significant increase, which would typically correspond to a tandem duplication or duplication of some sort of the gene for germline samples. And then the output of this caller, which then detects sort of using window approach, if typically the exact endpoints are not known, if you're using a panel or exome, clinical exome chemistry, because the intronic regions where often these endpoints fall for these chromosomal region deletions or duplications, don't have data for those non-sequenced areas of the genome, so often there's a windowing approach that helps determine across an exon or part of an exon, is there a higher or lower signal that is significantly different and consistent with a change in the overall chromosomal copy number of that region. So those baseline samples, the algorithms in the secondary analysis then produce these copy number calls, which are then sent on, for example, in this case, BRCA1 copy number variation, exon 22 deletion, from the upstream secondary analysis software through VCF to QCI Interpret via, in this case, upload to QCI Interpret tools. So there's API integration that can get samples to QCI Interpret directly. So then this gives you a first glance at some of the QCI Interpret how copy number variants are displayed. So if they're exon level, it'll tell you which exon in the gene is deleted or duplicated, for example. It'll show you the relevant ACMG CMV classification in the assessment window at the top when you're looking at the details of a variant. And then there's this bibliography feature, which when you click on that, it brings up the supporting literature for the particular variant you're looking at as well. And this assessment is also computed and shown in the top and right in terms of what it nets out. Is it likely pathogenic or benign or what have you? It sort of provides that visibility immediately up front in the top of the window. So touch on a few more aspects. The copy number variation can be quite complex. So I want to share a little bit about some of the things that QCI does to simplify it for you and streamline your interpretation workflow. So one of the needs is there can be multiple exons called within the same gene. So imagine, if you have whole genome data, for example, and maybe on the left side of the slide here, you might have a call, since you have genomic data for every base in the genome conceptually, it can detect the precise endpoints of a deletion, let's say. So in this mock-up on gene 1, you have exon 4 and 5. It may have, part of the intronic sequence to the left of exon 4 to a little bit of the intron sequence at the end of after the exon 5, and it can know that whole chromosomal range is missing because it just sees the junction between to the left of exon 4 and to the right of exon 5, and it can call a copy number loss with precision. And that's fine. Folks doing whole genome sequencing with structural variant callers that produce input like that are fully supported. In QCI, that would just be displayed as the exon 4 to 5 deletion, and that enables clarity in terms of the span of these large copy number alterations. Most people, though, are using panels or clinical exome sequencing, and there you don't have intronic information. You don't have any data for the intron. So you just have data for the exon, typically, sometimes the UTR, sometimes on the promoter, depending on the assay design. But what's important here is the copy number callers, if they're calling at an exon or region level, they often will be exon specific. And if conceptually you have, let's say that exon 4 and exon 5 are deleted, from a panel data, you'll only see, you'll get two different variants in the output. A call that exon 4 is a copy number loss, and a call that exon 5 has the copy number loss. But there'll be two separate variants rather than one, as you'd get from a whole genome sequencing. And so QCI handles that automatically for you. If the variants are adjacent in the gene, it'll automatically determine what the corresponding actual biological deletion would be, and likewise indicate exon 4 to 5 deletion or amplification, whatever may be appropriate. So it sort of coalesces adjacent exons for you, so then you don't have to do extra work in producing your final reports, for instance, or do that work yourself. It sort of takes some of that burden of that increased resolution from you, so by automatically grouping adjacent exons that are deleted or amplified together. Next part here is that pathogenicity classification, which again is done automatically based on the current available evidence to QCI, and it's done again with full transparency. So many good ClinGen slides, paper, and so forth on the Cmv guidelines. So I won't go into depth today, but just in the interest of time, there are some core differences. So it does have the benign pathogenic categorization, but instead of the ACMG guidelines that have these different levels of supporting evidence, from very strong or moderate, so on, supporting, instead the CMV guidelines use a points-based system. So for different criteria here, for example, it's a single to multi-exon deletion in an established haploinsufficient gene, in this case a DMD variant we're looking at, and it disrupts the reading frame and is predicted to undergo nonsense-mediated decay. That's in these guidelines criteria 2E6, and that gets 0.9 points, for example. The way they work is you sum up the points over all the different criteria that are applicable, and that overall sum is then mapped into the category of pathogenicity. So QCI does this automatically for you. We already know, for example, all the different structures of these genes and determine, whether a particular deletion you observe is in the UTR versus exonic, or is it an exon that matters? Does it cause nonsense-mediated decay or not? Is it in a haploinsufficient or loss of function gene, and what that gene is? We sort of use both ClinGen's curation, but also supplement that based on the Kaijan knowledge base and the curation we've done as well. So it's all done automatically to apply those guidelines to create the sum, And that also is available beyond this assessment section in the computed classification at the top. You can sort of just click on that little eye icon and you get this view on the right that sort of likewise summarizes that evidence. It's important to note that QCI's Interpret is a clinical decision support tool. So it shows you the evidence. You can go to the bibliography, see additional literature evidence and the different criteria. It specifies in line, like it's, in this case, the Duchenne muscular dystrophy gene is known to be haploinsufficient for ClinGen. That's why it puts that in line in the explanation. But since it's clinical decision support, although we provide the initial classification, you have complete liberty to adjust it as you see fit based on your own expert judgment and review. So you can either use the classification or set the assessment as is based on your view. You can add a rationale on the right. If you have multiple people in the lab, you want to add a note about why you added or removed a criteria. But you can overall set the assessment based on and set a comment summarizing the evidence that led to your final assessment there. And can also indicate through reportability where on the report it goes. Is it been assessed? You don't want to put it on the report, or is it been assessed a secondary finding, for example, or is it a reportable finding that should go on the top of the report, so to speak. So just go in a little more depth about CMVs with curated literature evidence, again, what we call variant findings. We want to make sure that you can readily see which of the many CMVs you may have in a case have literature evidence. And the way we do this is through this variant table you see on the bottom right here, which lists all the variants, and the default sorts them based on pathogenicity, for example. And so the variant findings, although in this case I think it's sorted by variant findings, the variant findings column indicates whether the Kaijan knowledge base has evidence specific to that particular variant that's shown. Each row here is a different variant. So in this case, it may be a multi-exon deletion in Duchenne muscular dystrophy gene that was detected in the NGS assay. It shows that there's 17 different pieces of evidence specific to that particular alteration. We also have a second icon here, which is this little blue plus that indicates there's what we call additional, so to speak, relevant findings. So we're trying to make a distinction here between equivalent, pretty much the same alteration that you observed in your patient case, and other CMVs that overlap clearly, but may require a little more review to determine if they're relevant for your case. And I'll show you how that's done in a moment here. But to explain how you determine whether some variant is relevant or equivalent, basically each variant, the variant you have in your patient sample, which has a leftmost chromosomal position and a rightmost chromosomal position, and if it's coalesced, it includes the inferred intronic region in between. Compare that to the CMV variant from the literature of the same type, like comparing deletions to deletions or amplifications to amplifications. That's what I mean by type. And that what we've done in our curation of the CMVs from the literature have likewise identified the chromosomal ranges for them. And so conceptually, what we use is a similarity score that takes the base pairs that are in the intersection of those two ranges, the variant in your case and the variant in the literature finding, into account, and divides that by the outermost leftmost position and rightmost position of the union of them. And that gives you what we call a similarity score. If they're identical left and right endpoints on the chromosome, they'll obviously have 100% similarity. If they're not identical, you can see in these bottom right here, there's three other patterns of the curated CMV from the literature overlapping the CMV in your patient sample, containing it or within it, or overlapping it on 5' or 3' end. Those also can give you similarity scores, in this case, in theory, all 50%. So beyond the similarity score, we also provide the span type. and you'll see in the bibliography feature, this helps you review some of these variants that we call relevant because they're contains or within or overlap. Whereas equivalent, what we say, if the similarity score is 90% or higher, that's considered equivalent for our purposes. So in the bibliography feature, as alluded to, there are these literature references, and we'll show you a demo in a moment here and give you more of a feel for how this all works. But in the bibliography feature, when you click the View Bibliography to see the Kaijan curated supporting literature and other sources appropriate for the particular variant you're looking at, what we do is provide not only the source article and what we call our variant finding, which, based on the particular article may indicate which particular exon range was deleted, the germline versus a tumor deletion, for instance, a particular disease association, whatever, they're all in details there, which we do for all findings, the small nucleotide variant findings, for example, too. This particular case happens to also be in HCMD, so it has this full HCMD note as well. But there's a massive amount of evidence we curate beyond what is in HCMD as well, I should note. And so what's provided here is this Cmv Criteria Filter on the top right. So that lets you further narrow. If let's say there's only relevant findings, there's no, so to speak, equivalent findings, that gives you a little more fine-grained means to identify which articles have evidence for a similar variant in a case where maybe that the variant you have in your sample covers it, sort of is larger than it, or is within it, is sort of smaller than it, or overlaps on the 5' or 3'. So it gives you a quick way to drill into that literature. Some of these variants have quite a bit of literature on them, particularly some of the hereditary cancer variants and variants like DMD have been well studied and well reported on for a while. Also in the UI, we provide a means to not only see the similarity score and at a glance understand which ones appear to be essentially equivalent versus relevant. You can click on these links to see in the genome browser the same view. So I won't go into that right now, but sort of the range there lets you see essentially are they the same size and on which end do they differ if there's some overlap as well. So with that, what I'll do is pause here and transfer over to you, Martin, so you can do a walkthrough and give folks a sense of how the application works in practice.
Thank you, Dan. I'm just going to share my screen. Okay, thanks, Dan. So over the next several minutes, I'm going to demonstrate how you can approach the interpretation of CMV variants in a case using QCI Interpret. But let's first start by orientating ourselves as we look at the analysis I have open here. There are three main pages in the analysis. The variant list page, which I'm showing right now, and the variant details and the review and report pages, which can be accessed by clicking on the links at the top of the page here. The variant list page lists the variants in the analysis in this main table. And by default, the variants are ordered by their automated pathogenicity classification. The variant details page provides supporting evidence for the selected variants that will help determine whether you want to assess a variant or include it in the final report. And the review and report page lists any variants that have been selected for reporting and is where the case is finalized or the final case report is produced. And we'll come back to these sections later. So for the purpose of this demonstration, I'm showing, the case I'm showing here is an example of a clinical exome from an affected 9-year-old female, as indicated in the top of the screen here, with an undiagnosed disease that is believed to have a genetic basis. The top summary section is showing information for the selected variants and includes the computed pathogenicity classification. And by clicking on this information icon, I can quickly see the summary of which ACMG criteria have been triggered to generate the computed classification of this variant. The variant table lists variants that have passed a filtering cascade if one has been applied when creating the analysis. And by clicking on the filter settings feature on the left-hand side here, I can see details of the filters that have been applied to this analysis and the number of variants that have passed each step in the filtering cascade. And there are a number of different filter types that can be applied to an analysis. So for example, here, I'm applying filters to retain high confidence variants that are not commonly found in the general population and are predicted to have a significant impact on a gene's function in the context, the genetic context of interest. And you can see by applying this filter cascade, I've filtered the number of variants down from almost 85,000 down to just over 500 variants that are of potential interest. If I go back to the main page, I can search this table further on specific keywords or criteria to quickly home in on the variants of potential interest. And the table also contains information specific to each variant. And this includes the phenotype associated with the gene, the mode of inheritance that is associated with the phenotype, the inferred zygosity of the variant, and known or inferred functional impacts of that variant, as well as the population frequency and the number of variant findings that are present in the Kaijan curated knowledge base. And there's also a link to HDMD if the variant is present in that database, which can be opened with a single click. Now, to begin reviewing this case, we can look at the variants in the main table. And I could start by further filtering the number of variants in this view by using the view settings feature on the right-hand side here. So for example, if I wanted to start with variants that were predicted to be compound heterozygous, or if I wanted to focus on copy number variants that were pathogenic, I could change the filtering here to do that. But since the variants are already ordered by their potential significance as based on the available data, let's just look at the variants in the top of this table. The first variant is in BRCA1 and is classified as pathogenic. And although this variant is reported to be dominated, it is for a hereditary cancer risk. And given that this is a child, the presence of this variant would not explain a rare genetic disease. So however, if this case were an adult, this could be a relevant variant to report as a secondary finding, which is a feature that is supported by QCI Interpret. Moving on to the next two variants in the list. Both are classified as pathogenic and both appear to be well studied based on the large number of findings in the Kaijan knowledge base. However, we can see that both variants are recessive and both appear to be heterozygous. So these variants are also not likely to be relevant for the disease in this particular case. But now if this was a carrier screening test, then of course these variants would be relevant to reports. Next, we have a variant, a multi-exon deletion that impacts exons 76 to 79 of the DMD gene, as we can see here. And we can see from the variant findings that there is a case with a reported copy number loss of high similarity that is present in the Kaijan curated literature. And we can also see by hovering over the blue cross that there are additional CMV variant references that, while of a lower similarity matching score, might also be relevant to review. And to review this literature, we can simply click on the View Bibliography feature here, which will open the bibliography for this variant. Now, in the bibliography feature, I am able to filter for references by the type of study in which I'm interested. And so I've selected clinical cases in this example. But I can also filter references by the Cmv criteria that are matching between the case variant and the reference, such as the type of overlap between the case and the reference variants. Or I could add a similarity score cutoff to filter the references. Or I could filter the references to only those, for example, that had precise endpoints reported. I can search for a specific publication in the search box here, or I can filter the references by the author. And I can also sort the references by similarity score. It's the default for a CMV variant, but I can also sort those by author or the date of the publication. And if I really wanted to quickly hone in on the variants that had a similarity match above 90%, I could click on the checkbox here, the show variant-specific references only, and that will now filter the references to only those that have a similarity score above 90%. Now, if we take a look at the reference type selected, we can see that the first one listed has a similarity score of 90%, as I mentioned, and that this the test variant overlaps with the reference variants. However, we can see that while the deletion in the case is the deletion of exon 76 to 79, the deletion in the reference is a deletion of exon 75 to 79. So the reference variant impacts additional genomic material in this case. Details about the genomic regions impacted by both variants are shown here, and clicking on them will display the selected region in a genome browser. And by clicking on the title of the publication, this will take you to PubMed, where you can view and review the abstract or open the full text article if it is available. So now, for example, if we look in the next variant in the list, we can see that this has a lower similarity score of 49%. And similarly, the variant in the reference starts at exon 72. So again, this variant in the reference impacts a larger genomic region than the variant in the case. Okay, so now I've reviewed some of the variant references in the variant table. I can return to the variant list, and looking at the variant details, we can see that DMD is both recessive and X-linked for the reported phenotype. And considering that this is a case for a female and the variant does not appear to be a homozygous loss, it is unlikely that this DMD mutation would cause the observed symptoms. So then moving into the next variant in the list, we see a deletion of the EP300 gene, which is also classified as likely pathogenic in this case. This variant is dominant in a rare disease gene, so might be consistent with causing the disease in this case. So if I now go into the variant details for this variant, we can review additional information that might be able to help assess this variant. The first section to look at is the assessment section. And we can see here details about the Cmv classification that have been triggered for this variant. So the variant is obviously impacting a protein coding gene with functionally important elements. Both breakpoints are within an established haploinsufficient gene, in this case, EP300. And these breakpoints delete the entire coding region of the gene. So this is equivalent to a classification of PVS1. And so the resulting score given to this Cmv classification is 0.9, which is equivalent to a likely pathogenic classification, as we can see here. So if we now go to the bibliography for this variant, although we don't have a reference which has a high similarity score, the highest being 16% in this case, we can see that the case variant completely covers the reference variant, and so may have a similar or more significant impact for this reported variant. So based on this, I could perhaps feel comfortable about adding this reference as supporting information for this variant. And I can do this by clicking on the reporting status icon here. And this will add the reference to the report if I do decide to report this variant out. Now, having added the reference of interest, I can return to the variant details page and look for additional evidence that may help further assess this variant. And if we look in the effect on protein section, we can see here that we have a link to Decipher. And this means that Decipher has an overlapping variant, overlapping with our variant of interest. So that means there's potentially relevant unpublished case evidence available. So I can click on the Decipher link, and this will take me to the Decipher webpage. And I can explore this evidence in more detail here. And we can see that not only are there a number of other unpublished case reports for CMVs in this region, but several of these are also reported to be pathogenic, the one shown here in red. And we can further see that one of the variants covers the region of the gene very similar to the region that we are seeing in our case. And this is also reported to be pathogenic. So having reviewed this evidence in some detail, I can now go back to QCI. And here I can now explore additional resources for the gene of interest. So by clicking on the link here, I see I can have link out to HGMD or OMIN or gene reviews. And after perhaps concluding that the variant's phenotypic spectrum is consistent with the suspected diagnosis, I can now confirm that I can now confirm the assessment and set the final assessment for this variant. So I can set this as being likely pathogenic and reportable, and I can do that by clicking the Set Assessment button. And here I have the option to add a comment to describe the interpretation, and this will update the assessment. So now that I've added the assessment, I can go back to the variant list. And we can see now that this variant has been reported out or will be included on the final report. Now, at this point, I could continue to review additional variants in the case, but once I'm satisfied with the assessments, I can then go to the review and report page. And from here, I have a number of options. I can choose to directly sign out the case, or I could change the state of the case to flag it for additional review or flag it for final approval by the laboratory director if needed. And finally, I could also generate a preview of the report. So clicking on this button will generate a PDF version of the final report if I wanted to do so. So with that final step, I will end this demonstration, and hopefully that has given you a good sense of how the new CMV interpretation capabilities and content that QCI Interpret provides can be used in your variant interpretation workflows. Thank you for your attention, and back over to you, Dan.
Thanks, Martin. Great, so I'd just like to summarize sort of what we shared today with you. It's sort of first and foremost, the QCI Interpret provides a scalable, standardized, with the guidelines, streamlined next-generation sequencing interpretation reporting tool. And what really sets it apart is this up-to-date integrated evidence that's constantly curated by the Kaijan scientific team that keeps your ability to understand what's clinically significant or actionable constantly up to date. And what we highlight in today's webinar was really the support QCI Interpret has for exon-level CMV interpretation. It's a lot more next-generation sequencing secondary analysis pipelines are detecting. And the content, as well as the guidelines here, really helps to put into biological perspective. So it enables you to quickly triage exon-level CMVs with the automated ACMG CMV classification guidelines. I should say not only exon level, but gene level. It also supports cytogenetic level CMVs if you happen to have those in your VCF as well. As Martin showed, it sort of gives you a means to see if there's unpublished evidence of similar variants in sources such as Decipher. And I should note that those links only show up when there is evidence for the same similar variant, overlapping variant in Decipher, for example. Only provide those links in those cases. To streamline your review. And then all this integrated literature, we have over 60,000 articles or variant references that refer to case studies of reported CMVs. And that lets you quickly identify which of the CMVs you have, even if there are variants of uncertain significance, have been reported in published cases in the literature, both through the HGMD integrated content we provide in Kaijan Clinical Insight Interpret, as well as for tens of thousands of reports that the Kaijan scientific team has also curated beyond that. And so that enables you to build up your case for assessing a variant as pathogenic based on both the ACMG guidelines and the curated evidence that's integrated as well. So with that, I'd like to thank you for your attention and take any questions.
Great. Thank you, Martin and Dan, for the interesting presentation and demonstration. So we have about 5 minutes left, and we can use this time to answer a few questions that have come in. If you have any questions for the speakers, again, please use that Q&A panel. And if we don't get to your question today, we will follow up with you by email. So the first question that we have is, how is the copy number content curated?
So there's two approaches there. One is HGMD, where one of the things we do is HGMD is another product Kaijan provides, which is a database of variants that authors claim are disease-causing, disease-associated. So the HGMD team curates the literature for those disease-causing mutations and compiles a database of those. Many of those articles are older, and so they didn't necessarily have any genomic coordinates. So one of the things that we did for the HGMD content is help translate or review and translate many of those references to genomic coordinates, which are necessary to match to your next generation sequencing data that likewise comes in chromosomal position range or genomic coordinates. That's a good amount of the content. A large amount of content also comes through Kaijan's continuous curation of literature. So one of the things the Kaijan team does is on a daily basis, it screens all the newly published articles for those that refer to commonly tested genes of interest, hundreds and hundreds of routinely tested hereditary genes, as well as all the somatic genes that are clinically relevant. And as part of that process of, whenever an article is found, there's different protocols that it can go through. But one of the things we do is try to also index variants of uncertain significance in these articles. And when we gather those as well, we include not just the reference, but for articles that talk about exon deletions and other kinds of copy number alterations, we map those to the gene and validate that the gene exon numbering is consistent with the current gene transcript, so to speak, so that we can then get it to genomic coordinates. And in genomic coordinates, that enables us to match within the application your user data, or so to speak, or the genomic data you upload to those alterations. So we have multiple, both the curation tool process to review that, also quality assurance along the ways to ensure that we match the, so to speak, the nomenclature used in these articles, which can be very variable. Sometimes they refer to exons, or sometimes they refer to ranges in genomic coordinates. There's not very consistent use of HGBS for CMV nomenclature yet in the literature, and so we take care of all that complexity in our curation process and protocols to provide a quite comprehensive view of what's known in the literature.
Great, thank you, Dan. I think we have time for one more question. Our last one is, does this include content relevant for cancer panels?
So the two types of cancer panels. For hereditary cancer, certainly. So hereditary cancer, looking at a risk for hereditary cancer, which many cases run through QCI are used to assess. Copy number variation in like BRCA 1 and 2 can account for collectively maybe ballpark 10% of the disease burden. So it increases the diagnostic yield for hereditary cancer to look at copy number variation. It's certainly relevant there. And pretty much actually not any particular indication so far, disease area where copy number doesn't play a role. And it's sort of ballpark, it can sometimes be 40% for some neurological disorders in terms of contributing to disease burden on a genetic basis. Sometimes it's a bit lower, but the key thing is copy number is relevant across the genome, also for hereditary cancer. For somatic cancer, we do curation for somatic cancer as well. It's not the focus here. The guidelines are focused on germline or hereditary disease interpretation, typically in the clinical exome context, but also appropriate for panels. For tumor samples, QCI also supports interpretation of them. We have different protocols that focus on overexpression of genes consistent with copy number amplification, for example, that will be picked up if you run a cancer sample through QCI, and there's appropriate copy number evidence in that context. So the bibliography in QCI will show information if you're looking at a pediatric tumor sample, for example, and you have a deletion in there. It'll show you whatever evidence we have in that, but generally it will be in a hereditary disease context, not a somatic context. And our focus really has been for the copy number content on germline copy number variation, but the QCI product supports both, and we get copy numbers from a variety of other sources for somatic interpretation as well.
Thank you. We're now reaching the top of the hour. I want to take a minute to thank our speakers, Martin and Dan, for taking the time to present this for us today. I also want to thank the audience for your attention. This concludes today's session on overcoming challenges of copy number variant interpretation with QCI Interpret, which was presented by Dr. Martin Jones and Dr. Dan Richards. We look forward to seeing you in another Kaijan Digital Insights webinar soon. Thank you, and I hope you have a good rest of your day.